Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we're joined by Kenny Workman, co-founder and CTO of LatchBio, to get his take on how the company is transforming data infrastructure for biotech solution providers. During the discussion, Ross and Kenny unpack why spatial biology and new assays demand scalable, high-performance data infrastructure, the 5 core components of the Latch platform and how they power a scientific insight, and the essential role of data in modern biotech innovation. Kenny also addresses the lag in technology adoption within the biotech industry and the importance of evolving approaches to data-driven research. Here we go.
Kenny Workman, welcome to the Data in Biotech podcast.
Thank you so much for having me. Appreciate it.
Awesome. Well, just to kick us off, would you mind giving us an introduction to your background and what brought you here today?
Yeah, totally. Have always had a fascination with living things for probably as long as I can remember. Like greenhouse, full carnivorous plants, like collection of frogs and newts, that kind of stuff early on in middle school. I actually studied EECS and BioE at Berkeley. Coursework was mostly math heavy with a focus on the underpinnings of machine learning systems. So statistics, linear algebra, complex optimization. This stuff, thought I wanted to head in that direction, but was also working in wet lab pretty much since I was 15. So started at local community colleges, transitioned to different groups in and around Berkeley, basic research in a variety of domains, lots of painful hands-on experience with rudimentary laboratory work, PCR, cell culture, Golden Gate. Around sophomore year, I transitioned from basic research into computer stuff, got an internship at a cell therapy company, working on both early machine learning and a lot of cloud infrastructure. Worked there while I was studying, and during the following summer, I worked at another kind of like SynBio company called Asimov, building computer-aided design systems for cell line engineering. And around that time was when we dropped out and started Latch.
Yeah, awesome. So that's a big decision. Sorry, how many years ago was this when you dropped out and started Latch?
Yeah, it's a good question. I was 21. This is 2022.
Okay.
At the very beginning of 2022.
Yeah. So
at the very beginning of 2021. Timeline
yes.
wrong.
Yeah. So a few years ago. So what was the insight that led you to and your co-founders to start Latch?
We didn't really have a deep and prescriptive insight into how biotech should be handling data differently, at least at first. Our early observations really those of repetition and waste. So my co-founders, Kyle, Alfredo, and I, we were all friends at Berkeley several years prior to actually founding Latch. And we spent many late nights hanging out, drinking, talking about all kinds of things, coursework, science and tech news, company ideas. Basically, I would riff on the cool things I was doing with computers at Biotech. And Alfredo at the time was working at Google and Facebook. And just talking about the gap in computational tools. So I was 21 at the time. They were a year older, and we just decided to send it and fill it. I also will note, I think many of these founding stories are over glamorized in American pop culture and suspect somewhat retrofitted to establish continuity with a unique insight that carried forward to current success.
Yeah.
And the only reason I bring that up, it's not to be contrarian or whatever. It's to highlight what Latch is not. Yeah, we didn't have a specific insight.
Yeah. So what did you do? I mean, obviously you've developed this platform over the last few years. So can you just take us on a tour of what that journey was like? Back to the beginning when you were just trying to, think about the intersection of computation and biology and try to figure out a place where you and your friends from Berkeley could make an impact.
So we had conviction in something more like a theme, right? That biotech primarily through data generation methods and a host of improved tooling was going from more of a science to an engineering discipline. Both to manage the volume of information, but also because new methods of discovery and engineering would emerge with scale of data. And so a lot of the industry would reorganize around computers. So we all understood computers really well. Most of us studied together at EECS. We met at the machine learning club and developed an independent interest in low-level systems, file systems, operating systems, languages. And we didn't really know exactly how to approach the beast that was the biotech industry. We just understood that this theme was playing out. We could solve technical problems at its intersection. and we just kind of approached every aspect of the company as an engineering problem. So particularly when it comes to like product development, it's like what insight do we have? We ran lots of experiments, collected lots of data, and redirected resources based on data. And that can kind of segue into how we started with conducting hundreds of user interviews, et cetera. But that is how I would describe Latch and why I push back against like you didn't have like a original good idea that is still a good idea.
Yeah. Well, it sounds like you had sort of a scientific approach to to like identifying the company that Latch was going to become. So tell us about those user interviews. Who did you decide to interview with, and what were the sorts of questions that you were exploring with them to try to figure out the direction you all wanted to head?
Looking back, it was really anyone who was doing biology or like programming at a biotech that wanted to talk to us. There was a crazy period where we were just like spamming every possible channel on LinkedIn and email. Got banned from multiple. Shout out to Bluebird Bio. Apologies for absolutely destroying your email servers. Like I have specific stories like this. Asking scientists like, "Hey, will you meet?" We had a limited amount of experience, right? I worked at a few companies, but we were just trying to talk, gather information. What stood out, and so like, very not focused, very not smart, but like ingesting data. What stood out was heterogeneity, and what I mean by that is like there were almost no patterns in the types of data generated, and the way that different experiments are strung together, combined, and the tools or infrastructure waiting downstream, and the number of types of teams waiting to interpret it to close like kind of the loop. Like nothing about this problem showed any sort of consistency from biotech to biotech at first. And I mean, there's definitely common threads running through the industry that I can see now. That just came 4 years after reading, conversations building, and 21-year-old me and co-founders and early engineering team were trying to connect dots prematurely. It was challenging.
No, that makes a lot of sense. So, can you give us an insight in like what were some of the early dots that you connected that sort of started the platform that you started to build back then?
Yeah, I can kind of explain what we built out, and how it encapsulates the quote, what I'll call like a skeleton of a generalizable like life cycle of data in biology. We definitely started with like the obvious and most computationally taxing pieces because it's like most aligned with the thesis of data generation like causing issues, and then like we encountered lots of biotechs who needed a lot more, and then we would either reject them or they would reject us, and we would just add a note to reach back out when we expanded. But in a nutshell, there's like kind of like five major technical components to the platform. Um, we started with workflows. So this is the most obvious, makes the most sense. It's like well-defined batch processing of a large molecular data. We then added like a place to store data, and this became a distributed file system that we wrote that kind of just ingests blobs of molecular data and then mounts it to the rest of the platform. Initially just workflows, but everything else. Uh, what became important next was, hey, you need contextual metadata, information about like the cell line you're using or the temperature in the lab that you need to associate in a structured way with files. So we built a registry, which is like a tabular database that scientists can interact with, but then can be pulled into Python. And that theme of like different levels of control, I'll touch on later. We then needed computers. Bioinformaticians need to write whatever code they want. So we built kind of a computing system called pods. And then over the last year, we developed kind of the creme de la creme, which is plots. And it's not just plots, it's actually the most ambitious thing we built. It's a combination of scientific notebooks and dashboarding with a custom kind of reactive framework and Python generated widgets that are backed by large computers. It's kind of like a graph pad for really, really large molecular data sets that can synthesize everything else on the platform. Very long-winded. You'll see this is a theme when I answer your questions. It's like I try to distill the complexity of what we do into something that is simple, but I feel like in that process it doesn't do what we do justice. And so I am often caught between like a rock and a hard place and trying to explain this stuff, I think.
Yeah, no, that makes sense. And you should feel free to go into whatever degree of detail you want to go into. That for our audience, I think we can handle the detail. So you built this biological data platform with workflows, data storage, contextual metadata, the ability to spin up and spin down compute for things that you wanted to run, and then and then now like a data visualization layer that's really high performance for like the large complex data sets that are being introduced. So, what is Latch working on now, and how does that relate to sort of like the customer discovery process that you've undergone throughout this whole process?
It's interesting. So in a nutshell, what we were working on over the past, I would say 4 years, is a infrastructure that stores, processes, and analyzes experiments. So ingesting streams of data coming from different machines or experiments, processing them into a state that's human readable, and exposing them, often layering or synthesizing them to different members of a biotech organization so they can draw scientific conclusions and make progress. I'm sure many of your listeners can relate, but rarely in early work did we encounter issues consistent with like the original thesis that the scale of data was delaying experimental cycles or straight up stopping them because existing software systems were unable to handle load. In like reality, like most of the problems in analysis with the largest return on the base of science come from basically team and project organization. So it's like modern biotechs are, I like to describe them as human engineering projects that need to coordinate very diverse teams around scientific consensus, but everyone has like some different training, speaks a different language, and key to analysis has like various levels of computational fluency. So like the example I tend to give is I'm grouping lots of things that shouldn't be grouped, but for the sake of this example, it will make sense. Immunologists, molecular biologists, chemists, they all need graphical components and click controls. Computational biologists don't really do well in like AWS. They need kind of a sandbox where they can write basic Python and R code and use their unique mix of a basic programming ability, but heavy statistics and like domain knowledge. Bioinformaticians and software engineers need to basically run whatever computer they want with their favorite tools. Otherwise, they'll like freak out. They'll lose control. So it's like in a single leg of a cycle where you're developing a therapeutic, you have to bring all these people together. And if you're building a platform where they're communicating, you need to like build product for all of them. So that is initially what we were working on and what problem we were trying to solve. And we had a modicum of success there. We've actually focused away from this in the past year. And I think the story there is pretty interesting. I'm so happy to get into that.
Yeah, but no, please. I'd love to hear, what drove that shift, and what the shift is toward.
So I think this is a pretty interesting and very much earned secret that only came from like navigating the edges of the industry and throwing things against the wall for years. But biotech companies actually make poor customers. There's a few things here, and some of them are perhaps somewhat controversial. Let's start with like the least controversial. Experimental structure between companies look very different. It's hard to establish this. So what this means for a cloud infrastructure company is finding companies that have unique challenges at scale of data is hard because of the heterogeneity. So it's just a consistent pool of customers. And any attempt to kind of like group these by therapeutic focus, disease, et cetera, in practice fell up short. Ecosystem is volatile. I think biotech's very unique in that R&D is very overrepresented in startups. And so like for data analysis products, like all the interesting useful problems are in R&D. So all you're working with startups, they fail a lot. Or they get merged, but then the merging company has no interest in working with you. So it just makes them challenging in that regard. And here's the most controversial bit. Compared to most industries, the dollar amount raised on average is exceptionally high, and the feedback cycles are quite long. So market forces are pretty weak, and this leads to what I've observed as very economically irrational behavior. I've seen teams burn millions building internal tools instead of running proper vendor evaluations because they could. And I saw many of these companies, these same companies, shut doors years later, where it's entirely unclear to me. I'm not saying I would actually never suggest that the reason was because of this decision. I would say that this decision is a symptom of a broader culture of vertical integration without need. But like no one knows when you see big Nuco going out of business, like that's some that I observed, and that waste never made its way into the public dialogue. So a huge part of this is like you have to understand, startup physics, like innate truths about your market when trying to build a company. And there's certain things you can and cannot control. We can control the technology. We can't control the market education about an entire group of companies and what they will and will not buy. So for all those reasons, we kind of moved away from biotechs. And if you think about it, the really gnarly problems come downstream of the new class of molecular measurement technologies that generate like actually big data, like terabyte per run data. You can think like single cell and spatial, epigenetic, proteomic assays. And this is where the need for data infrastructure was like high and like actually urgent. So because the biotech customers that did adopt us, they're I will say we did have early success with biotechs. There are some biotechs that are absolutely on the forefront of the adoption of new technologies and have this need, but repeatedly finding them and ensuring that we would have consistent usage. Their compute usage was spiky and moved up and down with the natural cadence of their experiments. It was hard to find more of them, right, for the reasons I said earlier. Um, so we just did what made sense to us. If you think back to the thesis of the company, we found the companies that were generating the data. We went right to the source, and these are the solution providers building the new assays themselves, and packaging them in like kits and services and distributing them to the industry.
Yeah, that makes a lot of sense. Can you give us some examples of, what those new companies are and what are some of the assays that they're bringing to market and sort of like the scale of data that's being created?
So we work with quite a few, and they all do really interesting things. I would say like the true data intensive scale comes with mostly with spatial. So in that camp, we have spatial epigenetics. Atlas Exomics is a phenomenal flagship example of this. They've built this technology spun out of Ron King's lab at Yale called DividSeq. So it's called deterministic barcoding in tissue. You basically take a section of tissue, stick it on a purpose-built microfluidic chip, and then flow molecular barcodes over overlapping X and Y coordinates, such that their code ahead of time and what channel they went in, and you can resolve them computationally uh, post fact. Um, and then, because these are just like generic uh, library adapters, the barcodes end up like being generic library adapters for NGS. You can extend this to like any NGS derived assay, including cut and tag and attack-seq, etc. That, as listeners may know, or epigenetic measurements that just happen to use some hack with NGS to like get the readout. Curio Biosciences does true single cell spatial by somehow, if you guys should read the paper, managing to get their barcodes inside the nucleus of a cell, providing that like compartmentalization. We work with a company called Broken String that doesn't do spatial, and it's actually not an example of scale of data, but they like provide kits to let people evaluate the off-target or just editing statistics of genetic editing proteins for all of the hosted companies that are building things there. And focusing on the solution provider bucket, the reason they make such great customers is they concretely understand the ROI of Latch. Like it drives down the kit repurchasing cycles by helping scientists understand data and scientific questions faster, so they can like rationalize, um, why they're purchasing it.
So if I understand correctly, let me just try to connect the dots here to make sure I'm understanding. So you've got these companies that have got like new primarily spatial but just high data volume assays that they're bringing to market that um require high computational load and new computational approaches combined together to get the insights that customers of these solution providers need in order to get the value from the assay or the kits that are being sold. And Latch is bridging the gap between the hardware providers that are providing the kits and the assays and the scientists, or whoever the CROs or whoever the customers are of the assay providers that are then trying to get value from the data that's coming out of there. Am I thinking about that right? Or where would you update that understanding?
Yeah, I would say that's exactly correct. It's B2B2B, and that sounds kind of fake. And at the outset, if you asked us like, "Hey, is that what you're going to do?" I would have said that makes no sense. It's stupid. But like just because of the amount of complexity involved there, but that just turns out that's where the highest density of problems related to data generation exist. Are the people distributing the things that generate the data, and the added advantage of they have these like economic models that package this idea of kit repurchasing is also beneficial, but also that they don't want to build two products at once, and I think they understand that viscerally. Um, so for a variety of reasons, that was a bit scattered. they just make a really interesting segment to work with, and are closest to like this original thesis we had as a team.
Yeah, that makes sense. So these are very different assays solving very different biological problems. So how does Latch, work with one of these new solution providers, assay providers to um, bridge that gap, to stand up the workflow that the end user needs?
It's a good question. So the advantage here, and this is why I think like the history of the idea of maze, if you will, is like unique and helpful. We originally built this for biotechs, and so like our intention is like was a full replacement for AWS or GCP or et cetera for a biotech. Everything you need um, from that like complete data lifecycle to like all the relevant controls to modify things, like having good dev tools that work with the various levels of computational fluency for the people like working on them. Uh, those were all things that already existed prior to us making this pivot. And the answer to that question is we kind of set up the domain practitioners within the solution provider to have what they need to encode their domain knowledge into the tools and establish their own feedback loops in a variety of like very specific ways. Everything from uploading the workflow, which is a pretty solved problem. We can get into that later. To having like pre-configured environments for customers to like write code for pre-configured like dashboards, which is very much not a solved problem, especially as the count like objects get really large. Like actually building intuitive browser-based applications that like are performant is a pretty challenging systems problem in 2025. Go figure. But the point being is like for each of these components, the solution provider controls it and has like some amount of internal uh, engineering that's dedicated to Latch. They treat Latch as their um, data infrastructure.
So if I understand that correctly, they have developers on their end who are creating sort of templates within Latch that their end users can then build on top of to get the insights that they need from the assays that are going in, and then Latch, um, you know, the entire Latch ecosystem comes sort of prepackaged with those templates for the end user when they receive the assay or the like the machine, the workflow, the kits that are generating the data as well. Am I thinking about that right?
Yeah, that's exactly right. And this actually caused us, as you can imagine, to really, really focus in on white labeling the platform and making the entire platform feel like not Latch and exactly what the solution provider wanted. Um, we did this for a few reasons. Like the first one is pretty obvious, but you know, each solution provider is a domain expert in their relevant branch of technology. They have deep expertise, extensive relationships, trust that we will never have. We want them to leverage their own distribution and be in full control over their brand. And then the other thing is like you have to make these folks happy from a productivity standpoint. They they need full control to make the changes that they want because like that just matches their pace of development. Um, and so like white labeling in that sense is like it's kind of psychological, but it's like they should feel like it's their platform, so they have the license to make those changes.
Yeah, that makes a lot of sense. Did you consider trying to get the Latch brand out in front of end users and sort of like get into internal biotech customers that way? Or had you already come to the conclusion that internal biotech R&D shops were not where you were going to want to focus your intention anyway, and so white labeling and making your preferred customer happy was just the best way to go?
I think it was pretty much the latter from the start. It's pretty clear if you dig in that like like Latch is powering things for like the power users that are like reading the documentation or like getting in the details. And I think that's like the correct division of responsibility because it lets the solution provider defer a lot of cognitive load and wasteful activity on like debugging things or like redirecting requests to our team. Um, so like for folks that really dig in, that split is clear. But yeah, no, it's an interesting question.
When solution providers develop develop these new assays, and they get to the point of thinking about, how are we going to enable our end users to get value from the data that we're generating, and they're looking at, what are we can build it, we can buy it, what are the options out there to buy it. What do you see as being the aspects of the Latch platform that are sort of driving the most value for your customers today, or just sort of like making them most excited to use Latch as their white label data platform of choice?
I really think we're first in class in this category, which is like a data infrastructure specifically for folks servicing their own array of customers. Like the alternatives really are to build in house, and there's nothing else, I would say. And so, like, when we did drill into why Latch, the layers of stuff over the past year we built on top of, like, this was already an infrastructure that a team of 100 scientists could use to analyze data, but now we've layered in like admin controls, kit packaging mechanics, like billing management. The lift of doing that and starting from scratch or even extending your current system, especially in the current climate where it's really focused folks to sharpen their pencils and be um, kind of like precise with resource allocation. I think that it's been somewhat clear. Um,
Yeah.
so yeah, it's like us versus internal teams, and then like the lift for internal teams is just getting greater as our team gets better at our job.
Yeah, that makes a lot of sense. I saw that you just released some updates to your visualization layer. So you have like an H5 viewer, and so the ability to explore these highly complex, high data volume spatial assays in the browser has sort of increased. Can you just introduce us to that feature and walk us through what are some of the parts of that problem that are hard, and where you think what Latch has done is most valuable?
So I think for a long time our team stayed away from digging into the details of assays, um, and especially spatial, because it just requires like writing a lot of custom code that's specific to the problem. Um, but actually, as we started watching things unfold a bit more, we realized that a lot of the structure in spatial was here to stay, and that as we focused, and I mean in terms of like file formats, like operations you want to do on the data, and the differentiation would be from like uh, slightly nuanced differences between solution providers, like the differences in their technologies that we could pull out and let them extend. But it was very much worth it for us to like start building closer to like the details of the assay. So spatial biology is like I think even from domain practitioners um, underrepresented in its scale. So we've been working with a lot of Cosmex data. And I think an example we wrote about a few weeks ago that was shocking to me when I discovered it is that a single run in 2025 can be close to a terabyte in total file data. And the number and types of layers are incredibly fragmented on top of that scale. Um, so if I remember correctly, it's like 450,000 cells. For context, like a 10x Chromium run produces like 10,000 cells. It's like 500 something million transcripts, multiple layers of high content diff images that each kind of measure a different fluorescent emission wavelength that kind of represents antibodies bound to different proteins. So you're layering transcripts, you're laying images with phenotypic information, proteins, you're laying transcripts. Wait, did I already say that? You're laying cells. So those three things on top of each other. And then the browser has to somehow, based on like the geometric location or like the area in the tissue that the biologist is navigating, fetch all three of those things from where they lie in a file somewhere, and then performantly send them back and layer them on top of each other, downsample them and render them. And the interesting thing here is like the like laws driving how these assays work just point to further gains in throughput, molecular throughput, and cost driving down. Um, Something like 2 or 3x every 4 years. If you look into it, these problems are just going to magnify. So all that to say is like that is a great case study of the type of systems engineering challenges that are uniquely here at with some of the solution providers. Like they really have issues with computers.
Yeah. No, that makes a lot of sense. And it strikes me as you're talking that these, like when you talk about like geospatial computing and you think about a tool like ArcGIS, there's this has been, sort of over time solved in like the geospatial world, but our Earth remains relatively fixed in terms of the nature of the nature of the shapes that you're rendering and the nature of the data that you're layering on top of it. But when all of these new assays are coming out, and the tissue samples that you're running are different each time, and the nature of the data that you're layering on top is different each time, and the volume is just increasing and increasing. It's a different type of problem. Did you take any inspiration from sort of the geospatial approaches, or how do you characterize the differences between what you're doing and sort of the other computational methods that are out there?
Yeah, by geospatial approaches, do you mean kind of like the tools that emerged around like processing satellite images,
Yeah,
mostly?
exactly.
So what's super interesting is um, a lot of early computational, like classical computational uh, results, either in algorithms or distributed systems, were motivated by molecular biology. And I promise I'll return to the point, but like,
No, please.
um, so the great example I gave is dynamic programming with the advent of like uh, early NGS, the Human Genome Project in 2003 with shotgun sequencing. It's a classic algorithm taught in any undergraduate course, is completely motivated by these problems. And then a lot of like early distributed computing problems like MapReduce, like fanning out data over many computers and bringing them back together, motivated by like spinning up sequencing search servers. Uh, this kind of dropped off around this time. And then the new problems that started like pushing the frontier of scientific computing became what you refer to as geospatial. What I understand is like satellite imaging
Yeah.
and particle collider, essentially particle collider outputs. So it's I don't get into this too much, but like a lot of the like file formats we use that repurposed actually for the machine learning revolution, um, that store large matrices of numbers, and then like kind of the ecosystem for like doing algebraic operations on those matrices of numbers came originally from bio, then transitioned to like what you said, and are transitioning kind of back. Uh, the point is that yes, we use like an archival image layered format called PM tiles. It's just like a specific example. I'm sure these things are fluid and will change, but like specifically built to render large maps performantly. And then we just kind of like looked at how it worked and then like repurposed it where necessary, like hooking it into Plotly, which scientists love, as an example. And then on the other components of like this specific system we built, use like DuckDB, which is like an in-memory SQL engine. Lots of like low-level uh, programming ideas and like how to move data around efficiently on a server. But yeah, these are some of the ideas that like went into making this browser, and I'm sure will change, but have like roots in that field that you mentioned, which is cool.
Yeah, no, that makes sense. And the fact that you're using sort of an in-memory analytical database like DuckDB, like backing up all of this just highlights sort of the way that you, by focusing on the compute layer, you can leverage the best tools that are available today, and sort of like stay up. And the nature of the problems that you're solving forces you to stay up on whatever whatever the new paradigms are in data or computing that can help you to provide the value to the companies that are producing all of this data. Thinking through the problems that you're able to solve for the solution providers, where do you see sort of the next frontier of challenges that are going to come up for these NGS and high throughput assays, the spatial assays, and the end users of those assays over the next, call it, I don't know, 3 to 5 years?
Yeah, the 3 to 5 year timeline, I think it's pretty simple and doesn't take like an incredible amount of like specific foresight. Like take those numbers I just gave you for Cosmex. They're pretty similar across like most of the competitive spatial vendors, and now just like double those numbers and then half the cost. So these are going to break almost every like constituent of the computer. Well, like some cases break, in some cases just like make actual concrete delays in experimental cycles from computation, and we'll just like demand a team that has like pretty deep knowledge of how to build these things to like re-engineer them for the assays. So like that is the prediction, but I don't think it's particularly like it just makes sense. It's
Yeah.
like a profound prediction.
You mentioned that it's going to cause, in some cases, it's going to cause things to break. In other cases, it's going to cause sort of delays in experimental throughput, and that there's going to need to be sort of a re-architecting of the way that computational biology is done in order to make space for for all of the data that's going to be coming through. Does the nature of R&D or the nature of like the way that biotechs do business change as a result of that? Or, what are some of the second order impacts that you would identify from that?
That's an interesting question. I would bucket that stuff like as second order impact. So like single streams of experimental data becoming hard to manage won't really have like the structure of the impact won't be as you described. But what will happen is in pretty much every field of science, at some certain threshold of data generation, the entire approach to reasoning about the system or manipulating it just changed. And so, like, it won't be because like CosmX or Curio just starts like generating 5 terabyte outputs, but because molecular measurements are so cheap and abundant that you no longer have to like do the weird hacky stuff we've been doing in biology, and you can just kind of like understand how systems work in a data driven way instead of using rational design and like small scale experiments that are like really perturbing small sets of molecules at a time, etc. Um, One thing I will say tangential to this point is that currently we don't see this, and that I'm in San Francisco. Where are you located?
I'm in Cleveland, Ohio.
Okay, so I'm currently in San Francisco. I spent a lot of time in Boston. I would say that today, and it's happening less as like the market has been as it is, but like uh, tech adjacent venture capitalists have long been bullish on data generation. I think we talked about this a bit prior, but like what we see in practice is that while this thesis is directionally correct, it's the same thesis that like Latch is pursuing. Um, the actual like deployment, the scale, and the fragmentation of like the machines generating all this data is like not quite there. Um, so like as it really ramps up, kind of some of those things I was alluding to will begin to happen, like data driven, model driven discovery and perturbation of systems. Um, but yeah, I mean that's something that all kinds of folks are working on.
Yeah. Well, what do you think is the bottleneck that's keeping this? I mean, I don't know. Maybe it's just my own news feed, but I see lots of news about how AI is transforming biology, and about how like the data that gets generated is going to be able to create these foundation models that enable us to solve problems that were never solvable before. And it feels like the solution providers that you work for and the types of computational methods that you're applying sort of like fit very neatly in that. And I'm having trouble squaring that with what you just said, which is that like the VCs are very much bullish on data generation in biology, but you're not seeing like the on the ground impact or adoption like accelerating like one might expect. So first of all, am I understanding that correctly? And if I am, like, why do you think that sort of contrast exists?
Yeah, and to be super clear, like I'm just suggesting there's like a lag. And again, the
Totally.
direction is totally correct. There's just a bit of a lag, and when it starts happening, it'll really ramp. I think people forget when there is something that like blocks a process, whether it be in the real world or digital, it blocks a process, and like the impact of that. And here I'm specifically referring to like computational bottlenecks due to the volume of data will be felt. The reason you probably don't see that, and this is another slightly controversial opinion. Again, like the advantage point that we have is pretty unique because I've seen the internals of hundreds of companies over 4 years. There's a huge gap between what is marketed and what is actually going on within biotechs, especially with respect to machine learning based platforms. Like the reality is that most of these companies are not working nearly at the edge of like developing models, and that is because they're not nearly working at the edge of generating data. It's like a second order problem, and that is because like they are still leaning on old guard rational techniques when rubber meets the road, because like that is what has the highest probability of success to fill a clinical pipeline and return dollars to investment, which makes sense. It's just the pitch that they gave was not that in most cases. But like, like a lot of these things like fell back to the conservative approach when funding dried up, and it was clear that you couldn't keep telling the story of a platform without data, and a lot of the platforms didn't work. These are very broad stroke observations. At the same time, there are companies doing incredible work here. The virtual cell point, like a virtual cell is basically like a language model that ingests uh, lots of well-structured molecular data to kind of like aggregate the statistics across different tissues or different, whatever the focus is, tissues, diseases. It's an aggregation of the statistical state of a molecular system so that you can predict like a perturbation in the presence of a drug, for example. Um, there's some incredible companies in San Francisco tackling this problem in a very prescient way. And most of the way you tackle this problem is like really innovate on the data generation side and like um, build platforms that generate high quality data. So yeah, but hopefully that, again, a little bit scattered, but hopefully that answers your question.
I think it directly answers the question, and it also strikes me that the sort of capital intensiveness and the investments that you need to make in the data generation infrastructure causes you to want to get the most you can from the historical capital expenditure you've made in the data generation infrastructure, and so there's always going to be a lag between the creation of these assays and sort of the adoption of these assays, just because of the scale of the expense that needs to be made. To your point, not just in the infrastructure itself to generate the data, but in the computational pipelines and tools that will need to be there to ingest the data.
Yeah, to that specific point, I would say it's a little more deep than a lag. It was like these companies never had the culture or the correct infrastructure in place to legitimately be a data-driven biotech. And there are companies that are emerging that are data-driven biotechs and have this culture. And I think it will begin to show in like very strong top-line clinical data from programs that are the result of these efforts. I don't know, like we'll see it play out. Like a great example of a company who's actually reinvented themselves here is Genentech's, I think GRED. Like what Aviv is doing there with respect to the scale of generation, the use of the data generated to drive unbiased discovery, and the integration of those wet lab systems with researchers and computationalists is a great example of, like probably the model that will drive us forward into an era of biology that is too hard to reason about in context of a human mind. And like that should be a good example for folks to look at. Very inspiring stuff.
Interesting, and all of this makes me want to get into some of the things that you say in your manifesto. So one of the things, so you seem to be calling for sort of the continuous mining of biological data, and the ability to better teach computers about biology. What do you think that means? Over the long term.
Yeah, it actually doesn't mean that much. So I will say we were young, we're engineers, and we wrote this like from more of a place of just like inspiration and like hand wavy romantic poetry than like any concrete idea that people should take seriously. I think at the time we're all systems nerds. We're all like reading and like studying the internals, like databases, programming languages, these things, and we saw a future where like if these themes continue to play out, you'd have to kind of rewrite all these components for biology to get more performance. Like seeing it in databases is quite easy, like at the storage layer, like the layout of sequencing data, and like the way it's compressed on disk should be like purpose built for biological applications. So those are the kind of things that we were thinking about at the time. In practice, like it was just like not necessary. I think that the one place where um, this idea does extend to very concrete efforts now is on the like reuse, repurposing of public molecular data. We've all established that like data generation is a bottleneck in biotech above anything you can do with computers, because computers are just like making what comes from the data interpretable. Uh, but that doesn't discount the fact that there is a lot of unused experimental information scattered across NCPI servers, like Geo, um, that really cover a broad space of diseases, tissues, unique patient identities, like that are just not being used because the amount of human labor that goes into structuring them and making them consistent is quite large. very, very recently, there's an entire cohort of solution providers. Their whole job is to organize or clean, curate, organize, and then sell data to biotechs. Um, and we've started working with a few of those folks. The older ones are Rancho and Bioturing, but there's a new crop, Neuromics and Pythio. We've started building tools for them. And very excitingly, kind of a tangent, but I think the advent of language models in technology broadly, but especially in biotech, has been overhyped, and a lot of the promises of their concrete capabilities overstretched. But specifically in the problem where you are ingesting papers, study metadata, and scattered author kind of raw files, and trying to make them into a consistently structured object with like a set of rigorous tests, we have found incredible success using latest ideas in language model engineering. That is the subject of like a full hour of conversation that we're basically equipping these data providers with to label data more efficiently, put it up on the platform, and distribute back to biotechs. In some cases, the uh, data from the original labs, the revenues from proceeds of those sales, we're trying to redirect back to the labs for basic research. So just a cool branch, and probably one of the threads from the original manifesto that we're continuing to act on.
Yeah, no, that's really interesting. So where do language models fit in terms of creating like a single object that allows to like test and reproduce, as I understand it, research assets? Where's the language model in the loop there?
Yeah, it's a good question. So if you focus on just single cell transcriptional data, just for the sake of focus, you can kind of break down like what the steps you need to do from, hey, you have like the raw unprocessed state to like you have a finished object that's like fit for use or like sale, into like aggregating all the information to downloading it, which involves trolling servers, but also like getting through paywalls, scraping paper information, getting subs, etc. Constructing account matrix, running quality control in the account matrix, transforming the account matrix, typing the cells in the account matrix at a level that like is correct, but also doesn't piss off immunologists because they have such a artisanal um, like body of knowledge around like what cell types are that are like even different between who you talk to, and then harmonizing metadata. So like typing and using controlled vocabularies to describe like things like tissues, diseases, etc. against ontologies that have already been built, like Uber, On, and Mondo, and CL, and these things. The language model comes in the account matrix step when you are trying to take the raw files from the author, take the information in the paper, and take the study metadata, and then basically build a Seurat or ScanPy object, like a matrix of numbers that adheres to conventions that are consistent across all the objects. Is the variable index, the thing that describes the genes, is using an ensemble ID. Is there a column that has a gene symbol? Are the counts untransformed? Is there a consistent sample ID? So basically conventions that need to be consistent that would require a person going in and writing custom code, because the author's researcher, there's zero incentive to do these things ahead of time. That's where language models shine. And in the past 6 months, trends governing their autonomous use of coding tools, reasoning about coding tasks, as well as like other ideas in like what it's called like data set eval generation. So taking like a task and then generating prompts for and tests for the model from data itself and having it iterate on those goals. We've used a lot of those great ideas and tailored it toward this very specific problem to basically, I wouldn't say solve, but drastically reduce the amount of human labor that goes into this problem, to the point where like I'm very confident we will have most of what's on Geo ingested and available to biotech companies within the next 12 months. And our team will be releasing a very, very large single cell atlas at the end of this year in collaboration with solution providers, but also using these tools. And then they can be, used in ways you would expect for the metadata harmonization and cell typing problems as well.
Yeah, that's an amazing use case for large language models. And it's funny, for as many times as I hear people talk about the sort of off the beaten path use cases of large language models, just the core use of text cleaning, standardization, converting unstructured data into structured data that can be translated into outputs that are standardized across different places is just a huge value. And that's a really interesting use case.
Yeah, and you have to think about like why it works, right? It's for molecular prediction tasks that need to build some model and learn statistics of like physics and molecular interactions. an incredible amount of both architectural changes, but also like domain knowledge need to go into building new models. So if you're talking about like reusing like general reasoning models who have been trained on text, like the exact substrate for them to perform are those of text manipulation and human reasoning. And there are very few concrete examples of those that actually move the needle in experimental campaigns. But the one I described is potentially one.
Yeah, that's awesome. Well, as we head toward the end, I just want to ask you like a few final questions. Is there a misconception that people have about biotech and the way that data is used in biotech that frustrates you, or ways that you think people should think differently?
I think a lot of my frustration comes from the pace of adoption of new technology in the industry. That really explains most things. That explains why, as we talked about, like data isn't moving as fast at first glance as you would expect. That explains why we didn't have success with a lot of biotechs. You can have other reasons for explaining that. Like I legitimately think that's the explanation. You look at a lot of current problems with American biotech, cannibalization of clinical market share, clinical assets by China, the amount of these companies like trading below cash, et cetera. I am here for this industry. I've been here in many ways since I was 15. I'll be here the rest of my life. Uh, we need to get our act together in terms of like adopting new things and experimenting and trying. And we're kind of relegating to doing what we know because it makes sense will not allow us to compete in a fast moving world of innovation, especially when the results have not been outsized. Not to get into another tangent, but like most of the drugs we've been developing just continue to balloon in cost with very marginal impact in their given disease area on survival, et cetera, because like the actual mechanisms that we're tackling in terms of disease are just exceeding the complexity and the reasoning abilities of like a human mind. We need to adopt new technology to make progress in the engineering of these systems. And that just like extends beyond software. But probably not the direction you wanted me to go with that question.
No,
But
that's
yeah,
absolutely
that is
absolutely a fantastic direction to go with that question. So
yeah.
thank you. Is there anything in particular that you're really excited about, either for Latch or for the biotech ecosystem at large, over the next 3 to 5 years?
Yeah, man, it's really these tools. You really study like any industry, and whenever the constituent tooling gets better, things start to explode. And computing, the analogy for me is always like the resources in a personal computer and what that did for developers. Like, there's an entire class of problems that I, as an engineer, didn't even have to learn about because I have 32 gigabytes of RAM on my Mac. The kind of like struct packing and bit twiddling and like getting things to work on a 64 kilobyte machine, et cetera. Like, it just let you focus on like new, interesting problems as resources became abundant. And I think you'll just find like many of our experimental approaches, many of our reasoning about how to design an experiment campaign approach will become restructured as data generation and the second order effects of that, like because everything in biology is like, kind of like a derivation of like the core readouts. Like, we think of like molecular interventions or molecular manipulations. They're always like a way of perturbing like the original like NGS readout, and that will probably continue to be true with new technologies. But like the way of thinking about all these things will just drastically change as data becomes cheap and abundant.
And for anyone out there who is, founding a company, either a biotech company or founding a company to serve the biotech industry, is there any advice that you would offer to people having gone through the journey that you've gone through?
No, everything is fake. No one knows what they're doing, especially a 25-year-old kid who has yet to IPO his company. So I think just take everything with an enormous grain of salt and develop like an internal reasoning mechanism where you can like evaluate things for yourself, and that's it,
I love that.
including everything I said here. Right?
So for people who are interested in learning more about LatchBio, where should they go?
We have a great website, latch.bio. Pretty active on socials. So we try to write about what we're working on in as much detail as possible. And especially if you're a new solution provider and you are getting off the ground, like building like basically the hub and spokes of the industry, the things that will let all these scientists measure their systems in greater throughput, reach out to me directly at kenny@latch.bio or go to our website and put in a request to talk to our team. we're mostly engineers we want to talk to.
Yeah, and for anyone interested in learning more about sort of the high throughput assays that Kenny's been talking about on this call, I can't recommend highly enough their Substack that provides sort of a survey of these assays and the way that they work. It's a fantastic resource for anyone who's out there.
Thank you, sir, and shout out to the whole team for that. It was particularly to Hannah, amazing product manager who's really been writing a lot of these pieces out of the lead. So it's a lot of her work. For
sure. So thank you, Kenny, for joining us today. Really appreciate it and look forward to connecting down the line.
Sounds great. Thank you so much for having me.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.